How are you everyone? Today's I discuss the topic is genetics of Bacillus subtilis. First, scientific classification. Domain is a bacteria, phylum is a Phronicoids, class is a Bacilli, order is a Bacilliaceae, family is a Bacilliaceae, and genus is a Bacillus, and species is a Bacillus subtilis. Next. Introduction of Bacillus. Bacillus known as hay bacillus and gas bacillus, and this bacillus was discovered by Christian Giffried Ehrenberg in 1935 and but rediscovered by 1941 by the German army in Africa. Next, characteristics of Bacillus subtilis. Bacillus subtilis is a rod shape and it has heavy flagella and it is facultative anaerobe and aerobe, and it is a 0.25 micrometer to 1 micrometer diameter, and it is a 4 to 10 micrometer long. And habitat. Bacillus subtilis found mainly found in soil, human fish, and marine environment. Reproduction. Bacillus subtilis reproduction by vegetative reproduction, and then transformation, transduction, and conjugation. It has no sexual reproduction because it has no gamete. Binary fission. Binary fission is a asexual reproduction by separating of the bodies into two new bodies. Firstly, this picture is binary fission. First, cell is replicate its DNA. Next, the cytoplasmic membrane elongated, separating DNA molecules. Then, next cross wall is from membrane is invagitant. Then next cross wall from completely. Then it from cell daughter cell. Next, transformation. Transformation is a process of reproduction. It is uptake by free DNA in environment. First, recipient this is recipient DNA and this is donor DNA, and lysis. This process is called lysis. Then it degrade. This DNA is degrade and it moves to the environment. Then then this bacteria DNA at this part is attached, and then next completely attachment, and this whole process is called transformation. And this transformation first observed by Griffith, and it is used genetic engineering for insulin incubation. Next is a transduction. Transduction is a process by which one virus transfers the generic material one bacterium to another. This is a bacteriophage, and this is a bacterial cell. Then next, this bacteriophage attack this bacterial, and next 3 and 4 cell create new phase including phase and host DNA. Next 5 and 6 transducing phases inside donor DNA. Next 7 donor DNA include in recipient chromosome due to the recombination. This process is called transduction, and transduction means transfer of portion of DNA from one bacterium another by bacteriophage. Next, conjugation. Conjugation is a process by which one bacterium transfer to the genetic material for direct contact. And bacteria containing the F plasmid are designated F plus cells, means male. Then F plus cells transfer DNA to an F minus recipient cell from means female. Next, formation of mating bridge. This bridge is called mating bridge. And conjugative tube. And then next, a single strand of the F plasmid break at a specific point and brings to move into the female bacteria, and this process is called conjugation. Next, Bacillus subtilis has about 4,100 genes. The complete genome sequence of Bacillus subtilis substance QB928 has 41,46,839 DNA base pair and 4,292 genes. Several non-coding RNAs have been characterized in the Bacillus subtilis genome in 2009, including BSR RNA. BSR means bulk segregate RNA. The next, FSR is a small RNA found in Bacillus subtilis. The next, the replication of termination protein is one of only two well-defined proteins known to be involved in arresting DNA replication frog. The other being a protein known as TRAS from E. coli. TRAS means termination utilization substance. RTP was first discovered in Bacillus subtilis and has been identified as a DNA binding protein of the wing helix family that forms the dimer of 29 kDa. kDa means kilodaltons. Next, termination of replication. Bacterial replication, like that found in Bacillus subtilis, consists of two replication frogs that travels in opposite direction around the same circular strands of DNA. Next, this replication frog being at the origin of replication and travel in clockwise and anti-clockwise direction. This is a Bacillus subtilis genes. This is origin. Then this and this side has two replication fork, and this side replication move to clockwise, and this side fork move to anti-clockwise. These are star side, and it has 6 star side. Then next, next is RTP structure. RTP is a DNA binding protein from Bacillus that uses a helix-loop-helix binding motif. Binding of RTP to the asymmetric B portion of the tar side changes its conformation into an asymmetric wing-up, wing-down structure. Then next, replication of mechanism. Debate over the mechanism of replication termination by RTP has led to a number of mechanisms being proposed. Original work on these questions was hampered by an incorrect structure of RTP beings. This structure was produced in solution and was shown to be a symmetric dimer. Then next, uses. Bacillus subtilis is commonly known as Bacillus natto and it is used for Japanese food natto. Then advantage and disadvantage. Advantages: no outer membrane, so very exudated protein. Then it can be used heterologous expression system. Then disadvantage: commercial adaptation and variety of proteins, difficult to manipulate than E. coli. These are references, and thank you. Thank you for the watching my video, and I hope uh, I hope my I hope you are guys helpful. So मेरा इंफॉर्मेशन अच्छा लगा है तो आप लोग ज़रूर लाइक कमेंट एंड सब्सक्राइब कर दीजिए. थैंक यू.